In this video, we will be discussing the molecular basis of inheritance. In 1953, Watson and Crick introduced their double helix model of DNA, or deoxyribonucleic acid. It had already been determined that the hereditary information was encoded in the DNA, and it is also reproduced in all cells of the body. DNA is important in directing all of the activities of cells and organisms. It is also copied during DNA replication, so it can be passed on to other cells, and cells can repair their DNA. Early in the 20th century, it was not known what the exact genetic material was. There was discussion about DNA being or nucleic acids being the genetic material, or proteins, so a number of scientists were doing studies to try to figure it out. Morgan showed that genes were located on chromosomes, but both chromosomes are made up of both DNA and protein, so it was hard to say which one was responsible for heredity or which one was the genetic material. So this role of DNA in heredity was first discovered studying bacteria and viruses. Frederick Griffith was one of the first people that studied DNA, and he found that DNA can transform bacteria from one strain to the next. And he was working with two strains of bacteria that caused pneumonia, one that caused the disease or was pathogenic, and one that was harmless. When he mixed different strains, he found that some of the harmless or non-pathogenic strains could become pathogenic. So he called this phenomenon transformation. Now, he didn't know what exactly it was that was transforming these cells, but he knew it was something inside the cell. Here's what his experiment looked like. He took living cells, which are called S cells, and they are pathogenic. And what he did was, just to do some controls, he injected them into mice, and the mice died. So he knew that those were the disease-causing cells. Then he took some R cells, which are the non-disease causing, as a control, injected those into the mice. Mice didn't die. So he took a solution of S cells and heat killed them to see if he could get rid of their pathogenic capabilities. He injected those into the mice, and the mice lived. But when he took the heat killed S cells and mixed them with living R cells and let that incubate for a while, he injected that into the mouse, and the mice died. And he found that in the mouse was living S cells. So what had happened was whatever material was inside the S cells had transformed the R cells to become pathogenic. Now, the next experiment that really did confirm that DNA was the genetic material worked with a virus called a bacteriophage. And they are viruses that only infect bacteria, and they're widely used in research. A virus is made simply of DNA and protein, usually. Here's what they look like. Here's the DNA inside, and then you see the body of it, which is all made out of protein. Hershey and Chase are the ones that showed that DNA is the actual genetic material using a particular phage known as T2. And they designed a really cool experiment that showed all of this. And they concluded that the virus injected DNA into the bacteria, and that's what provided the genetic information to the bacteria to make more viruses. So let's take a look at that. They used radioactive sulfur and radioactive phosphorus for a couple of reasons. One, sulfur is only found in protein and not nucleic acid, and phosphorus is only found in nucleic acid and not protein. So what they did was they radioactively labeled cells that they're working with and the viruses. The radioactive part in this first experiment was the protein. So they allowed the radioactive viruses to infect bacterial cells. Then they cleaned them off by running them through a blender, and then they centrifuged them to separate them, and they found that the radioactivity was only in the new phages. What happens when the phage injects its DNA into the bacteria is it turns the bacteria into a virus factory or a phage factory. So then when they made the DNA radioactive, did the same experiment, they found that all the radioactivity was now inside the bacterial cells in the form of DNA. So they confirmed that DNA was, in fact, the genetic material. Once that was determined, the race to figure out what the structure of DNA was on. And the first couple of people who worked on that were Maurice Wilkins and Rosalind Franklin. And they used a process called X-ray crystallography that takes pictures of the molecule using X-rays. And so Rosalind Franklin produced a picture of the DNA molecule using this technique. Here's Rosalind, and here's the picture of the X-rays as they are diffracted through the DNA molecules that have been crystallized. Well, her X-ray crystallographic images were shown to exhibit that DNA was a helix, meaning a spiral. So Watson and Crick got these images and they determined that the DNA molecule was a double helix. And so they're given a lot of credit for discovering the structure of the DNA molecule, even though Wilkins and Franklin did the majority of the work. Here's what a double helix looks like. You can see it's two strands wrapped around each other. And then notice that the DNA is made up of a phosphate sugar backbone with nitrogenous bases in the middle. We've seen nucleic acid structure before, so this should be familiar. Remember that the bases, or the nitrogenous bases, are bonded by hydrogen bonds, and that's what holds these two strands together. So you can see the double helical structure. There are a variety of characteristics of the DNA structure. One is the double helix, another is the sugar phosphate backbone, another is the complementary base pairing. Remember that A and T only bond and G and C only bond in DNA. Another characteristic of the structure is that they are anti-parallel. That means that each strand runs opposite of the other. We'll discuss this more in class so it makes sense. And then again, Watson and Crick figured that the base pairing was complementary. And we've already seen this before. Here's the close-up structure of those. A and T binding together and C and G binding together. Okay, now the structure of the DNA indicates that it could possibly replicate itself, because if you notice, the sequence of bases in one strand is complementary to the sequence of bases in the other. And since complementary base pairing is the rule, only the complementary base can be paired up with the one base from a separate strand. So DNA actually is a template for itself. So in DNA replication, what happens is the parent molecule unwinds and the two new daughter strands are built based on base pairing rules. Here's a simplified version of that. Here's the parent molecule. We've got ACTAG, TGATC. Well, if we split those apart, the only thing that can fit to either side is the complementary side. So notice the sequence of this molecule is the exact same as the sequence of the two new molecules. We also say that DNA replication is semi-conservative because in the resulting strands after they are copied, there's 1/2 original DNA and 1/2 brand new DNA. So only half of the original DNA is conserved. That's why we call it a semi-conservative model. So let's look at replication a little bit more closely. It is very quick in its speed, and it's also very, very accurate. And there are a bunch of different enzymes and proteins that are involved in DNA replication. First, DNA replication gets started at what are called the origins of replication, where two DNA strands are separated. Usually this happens in many places on a given DNA molecule. So DNA is copied all along its strand, and it's going to proceed in both directions from each origin until the entire molecule is copied. If we look at that here in a eukaryotic cell, there are going to be areas that open up, and this can happen because of the hydrogen bonding is easy to pull apart. So then other molecules come in and they start replicating the DNA, and they work their way out until you get two new DNA molecules that are exactly the same in sequence to the original. Here's another picture of that. Now, the enzymes involved in this copying are called DNA polymerases. And there's a variety of things that they do, but the primary thing you need to worry about is that they copy DNA, and that it happens fairly quickly in eukaryotes and in bacteria. Here's a picture of all the different enzymes that are involved. Here's DNA polymerase. There's a bunch of others, one that holds them apart, one holds the strands apart, and others that bring in the new nucleotides to add onto the growing strands. Once DNA is copied, it's also proofread. So there are a variety of enzymes that are involved in this. What they do is the polymerases run along and they proofread the DNA. If there's any sorts of mismatches, other enzymes will come in and cut those out. So if there's any errors in base pairing, they'll be clipped out. Okay. Also, if DNA is damaged by harmful agents from the environment, other enzymes can come in and perform what's called nucleotide excision repair, where they cut out the damaged parts and replace it with new pieces of DNA. Here's how that happens. So here's a damaged piece. The nuclease, as it's called, is going to come in and cut out the damaged part, fill it in, and then DNA polymerase and ligase are going to fill it in and close everything back together. Change happens in DNA no matter what, and it's very, very important for evolution because in some cases, mutations or changes in the DNA cause genetic variation and can also cause phenotypic variation, which is what natural selection operates on when it comes to evolution. So some of these phenotypic changes may be beneficial, they may not be beneficial, and so their natural selection is going to act on them to keep them in the population or remove them from a population. Now, we've talked about chromosome structure before, and remember that a chromosome consists of DNA as well as proteins. Bacteria have a double-stranded circular single molecule or chromosome. However, in eukaryotes are usually multiple linear chromosomes made with DNA and other proteins. Okay, in a bacterium, the DNA is supercoiled and bound really tightly, found in a region called the nucleoid. In the eukaryotic cell, DNA is combined with special proteins called histones in a complex called chromosome or chromatin. And the chromosomes fit into the nucleus through a multi-level system of packing. Here's what that looks like. Here's the double helix. It's going to be wrapped around histones, which are then going to further be coiled and then be supercoiled into these really nice, tightly arranged chromosomes.